In 1665, the English polymath Robert Hooke placed a thin slice of cork under a lens and observed a highly organized pattern of geometric, honeycomb-like pores, labeled here as A and B. He called these small rooms cells. But Hooke was actually looking at dead cellulose walls. He entirely missed the living interior. He missed the active biology because he was constrained by his tools. His newly designed compound microscope and separate spherical glass lighting apparatus represented a leap in design, but severe optical distortions limited what he could resolve. 9 years later, Anton Von Leeuwenhoek built single-lens microscopes capable of magnifying objects up to 270 times. Looking at pond water from his desk, he observed moving, single-celled organisms he called animalcules, proving definitively that the microscopic world was teeming with active life. Modern biology reveals that in multicellular organisms, cells do not sit statically in a tissue. They operate as units that progress through a continuum of 4 highly regulated developmental states. To see how a body builds and maintains itself, we have to map this developmental continuum, starting at the unspecialized stem cell. The definition of a cell extends beyond its physical boundaries. It is an entity capable of shifting its identity to build and maintain a complex organism. The first state is the undifferentiated cell. These unspecialized progenitor cells utilize a mechanism called asymmetric cell division. When dividing, one daughter cell loops back to maintain the stem cell pool, a process called self-renewal, while the other commits to a specific lineage, branching forward to specialize. This branching cell possesses potency, or developmental plasticity. As it moves forward, its options restrict. It scales from totipotent, where it can build an entire organism, to pluripotent, and eventually funnels down to multipotent, where it can only form a restricted range of tissues like blood or skin. Meanwhile, that first self-renewing cell escapes cellular senescence. It expresses high levels of the enzyme telomerase, which protects the ends of its chromosomes and grants the cell immortal DNA replication. The branching cell eventually reaches state 2, differentiated. It acquires specialized physical features, like elongating into a contracting muscle fiber. At this point of terminal specialization, it permanently exits the active cell cycle, locking itself inside a resting condition known as the G0 phase. Every somatic cell contains the exact same genome. To achieve its specific physical shape, the cell relies on epigenetic silencing. It tightly knots unneeded genes into dense, inaccessible bundles called heterochromatin, while keeping its lineage-specific genes open and accessible as euchromatin. To gain advanced physiological function, the cell dismantles its replicative machinery. Biological specialization requires the cell to permanently sacrifice its power to divide and adapt. This strict specialization presents a mechanical problem. If mature cells are permanently locked in the G0 phase, how does a salamander regrow a completely severed limb? The answer is state 3, de-differentiation. In response to extreme trauma, mature cells undergo epigenetic reprogramming. They strip away their specialized structural features, revert to a generalized shape, and regain the capacity for active cell division. In highly regenerative amphibians, localized muscle, cartilage, and skin cells de-differentiate at the amputation site. This newly formed mass of progenitors rapidly multiplies, creating a dense tissue cluster called a blastema that physically rebuilds the lost limb. Plants execute a similar process. When wounded, specialized parenchymal cells lose their vacuoles and rapidly divide into a cell mass called a callus. This reaction is explicitly driven by the wound-induced transcription factor Wind1 and precise hormone balances. This capacity for mature cells to revert their specialization serves as an evolutionary failsafe. By abandoning their mature identity, an organism can utilize these unspecialized cells for survival and catastrophic healing. Entering state 4, redifferentiation, hormonal guidance assigns these cells new specialized functions. Connecting the states reveals development is a continuous cycle of specialization and reversal. This summary grid maps these states against their cell cycle, epigenetic condition, and biological examples. The resilience of a living system relies on the ability of its cells to dynamically manage their own developmental timeline, shifting states as needed to adapt, repair, and survive.